Good morning, everyone. This great start with Dr. Agres and Victoria's overview of the symposium. Let's just move on, and I will ask Henrico Langeveld, our first speaker, to share your screen. Henrico. Meanwhile, I will say that Henrico is currently a master's degree at the University of Pretoria in South Africa as part of the Malaria Parasite Molecular Laboratory M2PL group led by the Professor Lynn Marie Bergcox. He completed his undergraduate degree in microbiology, majoring in biochemistry, after which he had the privilege and honor to join this excellent group led by Professor Lynn Marie, where he completed his honors with degree and distinction, and during which time his passion for science and especially for parasite biology grew. The floor is yours, Henrico. Welcome, everyone.
Thank you very much. Thank you very much, Emma. So good day, everyone. Thanks for this opportunity to share some of the exciting results I've generated thus far. So our lab is interested in understanding the specific processes in Plasmodium falciparum in an effort to provide more detailed biological information of the parasite to support ongoing drug discovery efforts in South Africa. We are particularly interested in the asexual proliferation of parasites, and two key questions we are asking related to the 48-hour asexual proliferation cycle is: what are the regulatory elements controlling progression, and can these be associated with classical cell cycle regulation, or are they unique? As an introduction to Plasmodium falciparum's cell cycle and overlaying that to asexual development, the association between asexual stage transition and cell cycle compartments are complicated. However, we do know that the parasite makes use of multiple rounds of asynchronous and endomitotic cell division cycles that occur in the absence of a G2 phase. In this context, ring stage parasites are typically classified in the G1 phase, where cell growth occurs as characterized by the transition into the trophozoite stage. Entry into phylogeny then marks transition from the G1 phase into S, with associated multiple rounds of nuclear division to give rise to daughter merozoites. This is quite unique in comparison to the canonical cell cycle model of mammalian cells, where the cell division occurs in a synchronous manner with well-defined cell cycle phases. In addition to this atypical appearance of the Plasmodium falciparum cell cycle, the regulatory mechanisms and elements governing the transition from one cell cycle phase to the next is largely unclear when compared to the cell cycle of mammalian cells, where there are well-defined cell cycle checkpoints governed by regulatory mechanisms, of which cyclins and cyclin-dependent kinases are the master regulators. One complicating factor of studying cell cycle progression in Plasmodium falciparum and challenging the identification of regulatory elements has been the difficulty to tightly synchronize malaria parasites to a single cell cycle compartment, something that is routinely applied to mammalian cells and which has enabled an in-depth analysis of the cell cycle. A standard method to study cell cycle regulation mechanisms was therefore required for malaria parasites to be able to synchronize the parasites to a particular cell cycle phase, reverse the block, and monitor various parameters of the emerging cell. Therefore, our group has developed such an enabling technology whereby mitogens in the form of polyamines that are essential to the parasite's development are removed through the treatment of DFMO. This has been successfully used in other systems and cancerous cells. DFMO treatment causes an arrest in early trophozoite stages of the parasite, which led to a 97% synchronized population of parasites observed at 22 hours post-invasion, with a less than 3-hour age window. This system is therefore a very successful system to tightly synchronize the parasite at what seemed to be a G1 transition point. This arrest could also be subsequently completely reversed without affecting parasite viability through the addition of putrescine, which acts as a mitogen. This therefore indicates that the halt in the life cycle is completely reversible and does not compromise parasite viability, and therefore has the hallmarks of a cell cycle checkpoint. In order to associate the life cycle arrest and re-entry with a cell cycle checkpoint, DNA and RNA synthesis were traced by cytometry. From this, we were able to show that the parasites halted at a G1S transition point after DFMO treatment and was therefore arrested in their cell cycle. But an addition of mitogens allowed parasites to continue with nuclear material synthesis, and the parasites clearly progressed into S phase. This data supported the fact that the system allows induction of a mitogen-induced R point at the G1S transition in malaria parasites. This is the first example of such a control in cell cycle progression. Moreover, this arrest is a highly specific event associated with mitogen depletion due to DFMO treatment and not a general stress event, as sometimes induced due to other drug events or nutrient starvations, as is evident from the genomic signatures. This system therefore allowed interrogation of potential regulatory elements associated with the cell cycle control at the G1S transition point. Whole transcriptome analysis performed on cell cycle arrested parasites and compared to parasites at different time points during re-entry of the cell cycle identified regulatory candidates, including several kinase family members. Of particular interest to us was the inclusion of a group of novel uncharacterized kinases that showed differential expression based on the cell cycle arrest or re-entry at the G1S transition. One protein with the code 5800 is downregulated at the cell cycle arrest and is a putative cell cycle-dependent kinase, where the other two proteins, 2100 and 4400, both show increase in expression under arrest, but are downregulated during cell cycle re-entry, with annotations associated with the BUD1 MAD3 for 2100 and a cyclin-like annotation for 4400. We subsequently aimed to investigate the importance of these novel kinases to the cell cycle progression in Plasmodium falciparum. To determine the essentiality of the selected putative kinases, the selection-linked integration targeted gene disruption system was used. SLI is based on a due selection for episomal uptake, followed by integration, as developed by the Spielman lab. The locus for all three genes was successfully amplified and cloned into the SLI TGD vector, and episomal uptake was confirmed via PCR on all three lines. These lines were then subsequently placed under secondary drug pressure for selection of integration, but no recovery of parasites were confirmed even after 4 weeks in recovery, compared to controls of non-essential genes. This phenotype was confirmed for 2 independent biological repeats, each in triplicate, which provided us with confidence to conclude that these kinases were indeed essential to asexual parasite proliferation. To delineate the contribution of these kinases to particular phases of the cell cycle and associated asexual replication cycle, parasite state distribution was investigated during integration selection. Interestingly, differential phenotypes were seen for these kinases with 5800 and 4400, both displaying a delay in cell cycle progression. These parasites both showed a halt in progression at the ring early trophozoid state in the first replication cycle compared to wild type parasites. Importantly, with these proteins having a CDK and cyclin-like annotations and the observed phenotype, it is possible that they do indeed play a role at the G1S transition in malaria parasites. In comparison, 2100 did not compromise the parasite in their first cycle of proliferation when compared to the wild type, but rather the second cycle, the parasites could not progress, indicating a delayed type death response. As this kinase again is annotated as a BUD1 MAD3 family member, it is foreseen that this protein might play an important role in the SM phase transition. Thus, in conclusion, 3 novel uncharacterized kinase proteins have been indicated to play an essential role in asexual proliferation, coupled with a proposed role at the G1S transition for the CDK and cyclin-like kinases, and a mitotic regulation for the third kinase. And by highlighting the essential nature of these kinase proteins within asexual proliferation, introduced new potential drug targets in the kinase inhibitory space. And by that, I would just like to thank everyone part of the N2PL group for their assistance and support, especially my supervisor, and then also all the funding bodies. Thank you very much for listening.
Okay, thank you very much, Henrico. So we're open for questions. I see that there are none in the Q&A chat, but I will ask then, is Henrico, is there any, just by chance, look at these proteins in the sexual stage? One, and the other part is that how conserved are these proteins to mammalian proteins? Like how feasible is actually to make these a target for therapeutics?
Thank you very much for your question. So the first part of your question, because we couldn't get into parasites after integration, we couldn't determine their function within sexual stages, but by using inducible systems, that is the ongoing plan for future projects and stuff, to incorporate these kinases into inducible systems and determine their function, and essentially in sexual stages. And then your second question, I'm not too sure how closely related they are to mammalian cyclins and CDKs. That is part of our ongoing research being done to determine if there are feasible drug targets and all of that.
So, Emma, Rubaiyat has a question that he...
Yeah, I'm reading. Yes, it's in the chat. Rubaiyat and everyone else, just please post questions in the Q&A chat, but I will read. There's a question here, Henrico. So you mentioned that you were not able to generate any knockout parasites. However, you show a piece of data where different parasite stages were quantified in knockout parasites. Could you please explain a little bit more? How did you do this experiment? Thank you.
Sorry, would you just reread the question? I just missed part of it.
So you mentioned that you were not able to generate any knockout parasites. However, you show a piece of data where different parasite stages were quantified in knockout parasites. Could you please explain a little bit more? How did you do this experiment?
Is it about this?
Yes.
So this was during the first 5 days of starting drug selection. We determined the parasite distribution while inducing during selection, the selection process, selecting for integration.
Okay.
There's I hope that answered the question.
Rubaiyat?
I think we should move on. There's one more in Q&A.
There's one more question in the Ben Lifner asks, do you think the delayed death phenotype could be related to inhibition of apicoblast replication rather than nuclear?
It could be. That is a very good point to state. We will be looking into that. Thank you very much for the that suggestion.
Okay. Are there any more questions from any of the panelists? Or no? There is another one question. Kevin asked, are the proteins having target site or homologous sites to any available drug?
Sorry, just repeat that question quickly. Have they been?
Are the proteins having a target site or homologous sites to any available drug?
We have not tested that yet, but yeah, that will be part of future research being done.